All right, part 2 of chapter 8, still looking at enzyme activity. So in part 1, you saw that temperature and pH can alter enzyme activity, but there's also ways to control for enzyme activity, not just just randomly affect how it's working, but a specific means of controlling enzyme. And one way is called competitive inhibition. All right, and what you see here in the top panel is competitive inhibition, where you have the active site of the enzyme, and then still this orange dude is the substrate. The substrate is supposed to fit into the active site in order to break apart and have the individual products. In competitive inhibition, there is a competitor that competes for the active site, and so it's the purple piece here. The purple piece fits just right into the active site and blocks the substrate from getting in, and so then the action doesn't occur. The enzyme activity is inhibited, and that's if you have more competitor than you do substrate. In this bottom part, you have just a very little bit, a small amount of your competitor, right, the competitor inhibitor, a lot of the substrate, and so the substrate is able to win the race and fit into the active site, and if this reaction were to be carried out, what you would see would be the two end products. Alright, non-competitive inhibition. Alright, this is still a means to control the enzyme activity, but it's not working at the active site. So here's the non-competitor inhibitor. It's not competing for the active site, so in this case the substrate can still bind. However, the non-competitor inhibitor fits somewhere else. And this binding, this attachment, alters the enzyme in some way that even if the substrate is able to bind in the active site, the substrate isn't, the products aren't made. Whereas in this case, there's no inhibitor. Enzyme is properly working. Substrate sits into the active site and the products are made. So two main ways of inhibiting would be competitive and non-competitive inhibition. And then there's also what's called allosteric regulation. Alright, and with allosteric regulation, you have an allosteric site, and in this case you have the allosteric inhibitor binds to the site, active site is able to be produced, alright, and enzyme activity is reduced. Alright, there's a little bit going on, but not a lot. Here you have no allosteric inhibitor. Substrate binds, you got lots of products. And then allosteric activation, this is an activator, binds to the enzyme. Substrate comes in, and now you have a lot of product. Alright, so the allosteric site can be both inhibitory or activator. Okay. Alright, then there's what's called feedback inhibition. Feedback inhibition slows down or can even turn off a pathway so that cells are efficient. Alright, so in this case, you've got this cycle here. In the first scenario, the furnace runs to create heat for a cold room. The temperature goes up to 78 degrees. Once the furnace or the thermostat senses that the temperature is right, the furnace shuts off. Alright, the heat slowly dwindles, and then once the temperature reaches 70 degrees, then the furnace turns back on. And so you have this process of feedback. Alright, in this case, the biochemical pathway runs to create a necessary end product. Alright, you've got this multi-step pathway. The end product are your gold circles here. And then the amount of end product from the pathway is sufficient to meet the cell's need. And so then the presence of this high amount of product, actually inhibits the enzyme early in the pathway, somewhere up here. Then once the product dwindles, when the cell has used it, alright, then the low amount actually stimulates the pathway to start up again. Alright, redox reactions. You may have seen a little bit of redox reactions in the chemistry module at the beginning of the semester, but redox reactions are a combination of oxidation and reduction reactions. They're called redox reactions because they typically occur together. And oxidizing agents carry out oxidation reactions. Reducing agents carry out reduction reactions. And so what you have is a reducing agent, alright, that has the electron. In the process of oxidation, there's a loss of an electron, and so the green circle is oxidized. In this case, you have an oxidizing agent that is able to accept this electron, so it gains an electron and it is reduced. Alright, so why is it reduced even though something was added to it? Well, the overall charge is reduced. So it went from a neutral charge to a negative charge, and that's because the electron has a negative charge to it. And so generally you don't just have oxidation and that's it, because then you would have floating electrons in cells, and that's extra energy basically. And that can actually be damaging to cells, especially if you get a lot of that extra energy. And so it's always good to have both the reducing agent as well as an oxidizing agent to be able to basically match up these reactions and allow for proper health of the cell. Alright, so in terms of making ATP, alright, there's different ways that phosphorylation can occur. Alright, and phosphorylation is the movement of the phosphate group. So remember, the phosphate can move from in the periphery, or out in the cell, bind to ADP to make ATP, and then that phosphate is broken off of ATP to release energy. And then where is that extra phosphate group going to go? Alright, so there's three different pathways or mechanisms, alright, to recharge ADP to ATP. First is substrate level phosphorylation. The second is oxidative phosphorylation. And the third is photophosphorylation. Alright, these are going to be used in different processes. Prokaryotic and eukaryotic cells can both use substrate level phosphorylation. Prokaryotic and eukaryotic cells can also use oxidative phosphorylation. And the photophosphorylation, only those cells that are photosynthetic would use this process. Alright, so humans would not use photophosphorylation because we do not directly use energy from the sun. Alright, so just be familiar with these three terms. Alright. So, little explanation. Alright, substrate level occurs when an enzyme transfers a phosphoryl group from a donor directly to ADP to make ATP. Alright, this is going to be high energy. Oxidative phosphorylation involves the collection of redox reactions. And with the oxidative phosphorylation, you're going to have an electron transport chain where electrons are going to be moved down from protein to protein to help fuel energy production. Now, here's where we start talking about, actually I think we're going to stop here. That's the start into the next section. So we will finish the process of enzyme control and ATP production, the mechanisms behind ATP production.